So our next speaker is Dr. Doris Wu from NIDCD. Dr. Wu received her bachelor's degree from University of Wisconsin and Stevens Point and master's degree from University of Southern California. And her PhD from the Department of Anatomy at the University of California, LA. She received her postdoc training at the Mental Retardation Center at UCLA and also Department of Genetics at Harvard. She joined NIDCD in 1993 and rose through the ranks at NIH and now holds the position of the chief in the Laboratory of Molecular Biology at NIDCD. She received a lot of awards, including the ARO Award of Merit. She also received the Mentorship Award. She served as the NIH Women Scientist Advisors and has published many papers on the topic of molecular mechanism underlying inner ear development. So as a sensory system developmental neurobiologist, Dr. Wu was drawn to the beauty and complexity of the inner ear structure. And although she has never received formal training on this field, she set out to understand the molecular mechanisms underlying the formation of inner ear. She and her colleagues have identified some of the key genes and pathways underlying the formation of this process, and the knowledge gained from these discoveries continue to provide the basis for hair cell regeneration research and help us understanding the etiology of hereditary deafness. And the title of our talk today is The Sonic Hedgehog Signaling Orchestrates Both Cochlear Duct and Spinal Ganglion Development. Let's welcome Dr. Wu.
Thank you. I like to thank the organizers for the opportunity to tell you something about what we have been working on recently. And let's see. Oh, good. Okay. We cannot hear without our hair cells. We also cannot hear without the ganglia that connects the hair cells to the brain. So we always think that hair cells and ganglia development is somehow coupled, but the mechanisms we know is still few and far between. For example, tonotopic organization, the most salient feature of the cochlea. We know both hair cells and ganglia has this tonotopic axis. We know that hair cells is likely to be established early in this axis. But what do we know about ganglia? Are they the passive follower, or they actually have forms tonotopy independently from that of a hair cells, from those of the hair cells, and they joined up? So there's a very interesting feature about hair cell development that we think is always associated with tonotopic organization. This interesting feature is that cell cycle exit of hair cell precursor. Oh, I'm having trouble with this thing, am I? Maybe. Okay, so cell cycle exit from the apex towards the base of the cochlea, whereas differentiation goes from the base towards the apex. This is a very unusual phenomenon because everywhere else in the brain, the first cells that exit from cell cycle are the first one that differentiate. And the timing of cell cycle exit determines the neuron's fate and positional identity. So this bizarre phenomenon makes us all think this has something to establish this functional pattern early on. So over the years, we have now learned what is the underlying mechanism for this unusual organization. And that is the sonic hedgehog, a well-known secreted molecule during embryogenesis. In the ear, it is expressed in a subset of cells in the spiral ganglion neurons. But what you see is over time that the expression becomes restricted towards the apical part of the cochlea. And this restriction of expression is thought to be guiding the differentiation of sensory hair cell. As proposed by my postdoc back then, Jin-Wang Bak, who is now a full professor at Yonsei University. And it was also proposed by Ziyong Liu, who was a graduate student with Zhenzhuo when he was still a graduate student. And now he is independent investigator at Institute of Neuroscience in Shanghai. So what they think is that as cells exited from cell cycle, they are being held from differentiation by sonic hedgehog. And as sonic gets restricted, then differentiation occurs. Why do they think such a scheme? Is because a couple of years before that, Betsy Driver and McKelly's lab have shown that sonic in vitro inhibits hair cell formation or differentiation. So when you knock out sonic, you would expect that what we saw is that cell cycle basically, this thing is, maybe I should point this way. It's either responding too much or not at all. That cell cycle exit would go from apex to base, and now you don't have sonic. So then hair cells differentiate right after. And I will just show you one piece of result showing in control. Sonic is basically in the mid basal region starting out. And in the mutant that you see that, my apologies, the hair cell marker is starting out in the mid basal. And you see in the mutant, there's a lot more hair cells in the apex, and it tapers into the base. And the cochlear duct is shorter, but regardless that the shortened cochlear duct and the hair cell phenotype, that the ganglia is also affected. And here is a medulla section of control showing you spiral ganglion labeled by neurofilament. And you see only here, the apical region of the ganglia is positive for sonic hedgehog. Now in the mutant, the cochlear duct, there's no spiral to speak of, and the ganglia is in this little nubbin of signal, okay? So of course, the question we ask is, what is the role of sonic in spiral ganglion development? And I want to do a quick summary of spiral ganglion development. It actually starts early at the otic cup stage, and the cells from this neurosensory competent region exited from the epithelium, coalesced to form the cochlear vestibular ganglia that quickly split to form the vestibular and auditory ganglia to innervate various sensory organs. So after the cells exited from the epithelium, the neuroblasts continue to undergo proliferation. before they become post-mitotic and mature to form process to innervate various sensory organ and connects to the brain. So the question we ask is, when is sonic required? And the other question is, given this restriction of sonic expression that is attributed to the timing of hair cells differentiation, what does this mean? Does it mean this is the same group of cells that gradually concentrated to the apex and then drives the hair cell differentiation as a result? Or they are completely different group of cells? And so we asked that question by using lineage analysis of sonic with the sonic Cree ER mice. And so if those cells are labeling the same group of neurons, so by lineage, you will see everybody, doesn't matter when you activate Cree, you will see always the same population of cells being labeled. And first, what we did was we activate tamoxifen throughout the development and of the spiral ganglion neurons. And you see in this whole mount and section, pretty much it looks like majority of the neurons are labeled. And based on the innervation pattern, we think that includes both type 1 and type 2 neurons. So now, when we look at, when we do tamoxifen at a single age, at embryonic day 10.5, you see the lineage label cells are basically located at the base. At E12.5, single dose of injection, now we see the concentration of label towards the apex and less at the base. At 14.5, now you see that only the apex are labeled. So right there, here, these lineage analysis is extremely telling. It tells us that these expression pattern cannot be representing the same cells because we get completely different pattern. Okay, and the fact is that the sonic expression in these neurons are actually transient, because otherwise, when you do a 14.5 time point injection, you would the basal nuclei should be labeled, but they are not. So that means sonic is transiently expressed at a certain stage of during spiral ganglion development. And here's a summary of the early lineage gave rise to neurons sitting at the base and later on at the apex. And this pattern is totally reminiscent of what's known about the birth dating of spiral ganglion neurons, according to Rubin's thymidine labeled data 50 years ago, that the early ones are at the base and the later born ones are at the apex. Okay, so the question we ask is, what stage is sonic being expressed in, right? So here is a comparable section showing an early neuronal marker, neuro-D, labeling the neuroblast, and neurofilament of a more mature marker. And you see these two patterns largely complementary. You see when neuroblast is strongly labeled with neuro-D, it's not expressing neurofilament. And then later on, when neurofilament comes on, neuro-D goes away. Okay, so we compare this expression pattern to that of sonic and the receptor for sonic, patch 1, which is an indicator of which cells are responding to sonic. Okay, so as you can see first, sonic is expressed sort of in this intermediate region, this yellow bracket region. between strong neural D and strong neurofilament. So we already know sonic is at a specific stage of development. On the other hand, patch is actually overlapped very well with the strong neural D patch, suggesting that these are dividing neuroblasts. So to test that, we did a thymidine analog labeling, injecting EDU and at embryonic day 12, and then harvest 2 hours later and ask who's dividing. And what we see is that the patch expression and the EDU label overlapped very well. But what's interesting is that the sonic domain is devoid of EDU, meaning that these cells are not dividing, not picking up the analog. However, if we do the same experiment, inject with EDU and wait a day before we harvest, and now we see the cells, the sonic domain is labeled as opposed to this. So this is kind of a summary of all the expression and the EDU data, is that sonic is expressed in postmitotic neurons or nascent neurons, and patch is in the dividing neuroblast. So as I said, patch is an indicator of who's responding to sonic, right? So the question is, what is sonic telling the neuroblast to do? Does it regulate the delamination, or is telling the neuroblast to divide, which is a known function of sonic in other systems? And that because of this schematic that I'm showing here, you see that sonic basically feeds back to the dividing cells. It makes us wonder what happened to the first batch of neurons. Maybe they don't need sonic, and maybe that accounts for the numbing of spiral ganglion neurons. So meaning that in the FOXG1 mutant, that it will be largely comprised of the early born neurons, right? Or alternatively, each of these cohorts are represented in that nubbin of ganglia. So which one of these possibility is true? Oops. Oh, I gotta point this way, maybe. That's better, yeah. So unfortunately, there's not a marker that we are aware of, or any marker that distinguish the early each of these cohorts. And so we have to go back to rely on cell cycle exit, the time the neurons are born, as a guide. But knowing that sonic could very well regulate the cell cycle exit. So whatever these green neurons may not be the same as the wild type, but nevertheless, that's what we did. And we utilize again the EDU. And in this case, instead of harvesting 2 hours after we injected EDU, we waited, let the dividing cells dilute out whatever they pick up. But the ones that show strong labeling are the ones that exited from cell cycle shortly after the EDU administration, right? And so these are our one result in wild type, that we injected EDU at 10.5 and then harvested at later. And what you see is that the labeling, the high EDU labeling, are located at the base of the cochlea. And if we inject EDU at 12.5, now we see the labeling at the apex. So these results are completely consistent with what Jerry Rubin have done 50 years, as well as Bernd Fritz's lab has also demonstrated that, okay? So now, the question is in the mutant. So again, I just want to point out it's hard to see sections, but this is the where the nubbin of ganglia is located. Okay, so we injected at 3 different ages. Okay, and if this is primarily comprised of early born neurons, you will only see labeling at E10.5 injection. That's not what we saw. Oh, sorry. You see in each of these stages, you see labeling, heavy EDU labeling. They are overlapping, but they seem to be somewhat restricted, but nevertheless, the key is that neurons are being generated in this mutant over the course of development. So in summary of that result, the neuron, this is not the scenario that we have. Rather, this is the one that all cohorts are being represented in this reduced ganglia. Are they represented equally? That I cannot tell. And but what this result suggests is that the sequential production of neuron as in the wild type are maintained. So the question is, if this sequence of production is maintained, does that mean these neurons carry with them the positional identity, like they know where to go? Meaning that, like when the green neurons are formed, do they know they need to go to the base, or they just like passively go to the base? Is kind of like the bottom line question. Okay, so unfortunately, to ask that question is too difficult to analyze based on this, because the mutant is too severe. So we generated another mutant using NeuroD-CRE, which activates in the neurosensory domain. So it's a little later, and you can see the cochlear duct is much milder than this FOXG1 mutant. And this is only about 30% reduction in length. So the question we ask is, in this NeuroD mutant, if positional identity is already conferred to the neurons at the time they are born, then we may expect first no phenotype, because even though there's less neurons, there's also shorter. So it just evens out. We will not see a phenotype. Or the neurons know where to go, and there's not enough, and they are sparsely located. Another provocative option is that they know where to go, and they home into where they're supposed to go. And as a result, we create these gaps. So which of these scenarios is correct? And well, what we found right away was that none of those, because what we saw consistently is always missing ganglia at the apex. So we know that, I'm jumping ahead. So you can see here that there's a total 30% reduction of ganglia that is distributed, but you see there's a disproportionately reduction of ganglia in the apex. So it tells us that none of this scenario is correct. However, when we look at neurons being generated by looking at the EDU label, we see that they are pretty much, in terms of neuronal production, they are always reduced, but they look relatively similar, about 50%, about 40%. It may be a little bit more for the apical ones, but only 10% difference. It should not account for the absence of neurons. So, but what we also observed is an interesting thing is that in the control, the highly concentrated EDU label, meaning that the post-mitotic neurons, are located in the control at this region, the apical side of the cochlear duct or mid-apex. However, in the mutant, what you see is that there is a shift of that heavy labeling towards the base, as shown here. So these results tells us that, in summary, that there is there's no fixed positional identity of these neurons, but rather they are kind of like filling a space. And when you don't have enough neurons, then it's an accumulative effect that you see in the apex. So this would argue against that there is a positional identity ingrained into the neurons at the time they exit from cell cycle. Okay, so I summarize very quickly that what we have learned is that sonic is functioning, main function is to signal each cohort of neuroblasts to expand. And this is what we would call as a transit amplification, meaning, as the name implies, as the cells transition from the neuroblast stage to differentiation, they undergo expansion. Okay. And finally, I want to end by saying that this model that we started out with, that restriction of sonic expression, drives the hair cell differentiation. What is the nature? Like, are they migrating or whatnot? So since then, we have done more 3D reconstruction. And then it's not as pretty as the other ones because this is a commercial software. The old software from NASA was no longer supported. But in any case, you could see the sonic domain basically stays in the same position. But because the neuroblasts, the amount of neuroblasts that are delaminating are less over time, and thus creates this restriction to the apex, and which drives the differentiation of sensory hair cells. And this is how those two important components of the cochlea are being coordinated. And with that, I'd like to acknowledge Loxam Wong, who is the postdoc, was the postdoc in my lab who single-handedly spearheaded this experiment. And this is my current lab that hopefully someday I'll talk about their work. Thank you.
Questions from the audience? You think there are a chance actually to look at the spinal, to look at the neurons in vestibules? When we looked at the, compare vestibular versus cochlear, vestibular ganglion are much bigger, almost twice the size of the cochlear.
Is that right? So sonic is not expressed, if you remember the 3D reconstruction, sonic is in a subpopulation of the auditory ganglia, is not expressed in the vestibular, and neither is patched. So this is a solely spiral ganglion phenomenon. Matt.
Doris, you mentioned migration, and I'm wondering both the cochlear duct and the spiral ganglion are shorter in the hedgehog mutant. So have you looked at migration, or are there roles or changes in migratory behavior that you know about?
Yeah, no, that's a really good question. I think that it seemed like the amount of sonic and the cochlear duct is always coupled. You have more severe mutants, no growth. And then as you go less and less perturbation, then they have longer and longer cochlear duct. And even with the neuro-D, there's 30% reduction in total neuron number, and there's 30%